bit mystified by the concept of phosphorylation. It's so critical in endocrine signaling sequences. So this slide is simply trying to remind you of some of the terminology and such that you may have been exposed to in your biology or chemistry course. Remember, when we talk about intracellular phosphorylation, all we're talking about is one molecule moving a phosphate to another molecule. In this case, because it's endocrine signaling, we're mostly talking about proteins. So proteins are long chains of amino acids. The amino acids in the protein that are typically phosphorylated are serines, threonines, or tyrosines. So that's all that means. Those are the most common sites of activation for phosphorylation or adding the phosphate. Be aware, we've used this terminology before. In this connection, you will see reference to kinases, phosphorylases, and dephosphorylases. Remember, ASE just means it's an enzyme, and all it's doing is that those enzymes are contributing to the movement, the transfer of phosphate from one molecule to another. Phosphatases, for example, typically remove the phosphate groups. In the literature, you often see the word activation, and by activation, we just mean adding a phosphate, giving that molecule additional energy, additional capabilities that it previously didn't have. And so a phosphorylation cascade, a phosphorylation chain or cascade would be me, the activated kinase, or the activated molecule in this case, simply transferring phosphates to the front row here. And I can set fire to as many people as I want. And so, and then as you catch fire, everybody else behind you is going to catch fire. The point is, that's called signal amplification. We'll look at a little animation in a moment. But these phosphorylation cascades are frequently amplifying, and thus you have the entire cell excited with metabolic activity that wasn't present previously. So that's all this is about. You can sort through that at your leisure. And we've talked about the possible consequences of intracellular signaling. There can be a change in membrane potential, depolarization or hyperpolarization. We can have this G protein or PLC sequence or even a tyrosine kinase sequence. Rarely, the hormone can actually act as an antagonist. And I know you already know these terms. I just want you to understand that they're relevant in this context as well. Be sure you know the difference between an agonist and an antagonist that we talked about when we talked about the neurotransmitters. Now, this is even more true than we discussed with neurotransmitters. Just a few simple points here I perhaps didn't state necessarily all that well in this paragraph. Remember, the endocrine response is going to be receptor dependent. The receptor determines the biological response. Therefore, the number, the density, the relative abundance of the receptors is absolutely critical. And because there are a finite number of receptors, there is a finite number of receptors in the membrane, we say that the response of the cell to the hormone is saturable and graded. All right, so the more receptors that are involved, the greater the response. That's why it's graded. And it can be saturable because if the hormone is binding all of the available receptors, that's it. It doesn't matter how much hormone is circulating in the blood, the cell cannot respond to any greater extent because there are no more receptors to bind. All right. Unfortunately, there is a little bit of a complication to this. I'm happy to talk about a few of these complications. One of the complications is that for most hormones, and this applies especially to steroid hormones, although I realize we're mainly talking about water-soluble now, but this applies more so to steroids than water-soluble. The maximum biological response of a cell to hormone binding requires only that a small percentage of the total number of receptors be bound. Sometimes on the order of just 2 or 3%. So in other words, again, if we're a cell, the whole room is a cell here, and each one of the seats is a receptor, we can max out the ability of the cell to respond to the hormone can be at 100% if only this first row of receptors is is occupied by the hormone. We say that all the other receptors are spare receptors, S-P-A-R-E, spare receptors. That seems crazy. What would be the biological design? Why would that be a useful approach to things? What would be the basis of that design? Why would I have so many spare receptors? Again, hormones circulating through the blood out there.
To give the cell the ability to elicit response even in a low concentration of hormone.
I think in part you're right. I think it enhances the sensitivity. It increases the likelihood of hormone receptor binding. But be aware of it. I just want you to. So all of these things are true, but it does kind of complicate our picture when we're arguing that we only need a small percentage of the receptors to be bound in many cases for the cell to reach its maximum biological response. It's true for most hormones, but it seems to be for the steroids, especially testosterone, for example. We're down in the single-digit percentages are all that need to be occupied. So that's what I mean by maximum response occurs without saturation of receptors. I want you to know what we mean by saturation. What is saturation? Yeah, all the receptors would be occupied, so you don't really need to have that to happen for maximum response to occur. And then on top of that, the other complication, we've already discussed this. Remember, we talked about pulsatile secretion and why that's important. Remember, these cells are dynamically, I'm speaking as if they all act the same. These cells are dynamically altering their membrane receptor densities in response to the circulating level of hormone. Why do they do that? What's the hallowed principle they're all attempting to maintain? Homeostasis. This is just a feeble attempt to maintain homeostasis. So, of course, if that's true, that's sometimes called mobilization. That both transporters and receptors can be mobilized. They can be taken into the membrane and taken out of the membrane. Taken into the membrane, taken out of the membrane. So that's just my point here. That doesn't mean that the cell has to make new receptors, send them into the membrane, destroy them, and so on. No, they're just shifted like soldiers back and forth from the front as needed through a variety of regulatory processes. Any questions then about these terms? Just a lot of terms I wanted you to be familiar with. So this is a typical example of a second messenger signal amplification cascade. You can think of this as a phosphorylation cascade, and all I want you to appreciate from this is once that single hormone, that single molecule binds to a receptor, as a consequence, by virtue of this phosphorylation process whereby there's one molecule has the ability to phosphorylate 2 or 3 other molecules and so on, there can be rapid, potent, and powerful intracellular amplification or magnification of that single binding event at the cell membrane.
Receptor proteins interact with signal molecules at the surface of the cell. In most cases, the signals are relayed to the cytoplasm or the nucleus by second messengers, which influence the activity of one or more enzymes or genes inside the cell. However, most signaling molecules are found in such low concentrations that their effects in the cytoplasm would be minimal unless the signal were amplified. Therefore, most enzyme-linked and G-protein-linked receptors use a chain of other protein messengers to amplify the signal as it is being relayed. In the case of a protein kinase cascade, one cell surface receptor activates many G-protein molecules. Each G-protein activates many adenylyl cyclases. Each cyclic AMP, in turn, will activate protein kinases, which then activate several molecules of a specific enzyme. The end result is rapid production of high levels of the final product.
And that final product could be anything from a polymerase that directly alters or modifies gene transcription. It could be a regulatory enzyme that determines how much of a product is secreted. It could be anything.
Would this process of activating multiple G receptor proteins happen in things like neural pathways that we learned about earlier, or is this hormone specific?
Well, the simple answer to your question is yes, because of course, remember, many of these catecholamines act both as neurotransmitters and hormones. But typically when we talk about signal amplification, if you think about it, as a general principle, this wouldn't work too well in neurotransmission because things would get out of hand pretty fast. So for the most part, unless we're talking about neurocrine specific pathways, this applies only to endocrine circuits. I did it again. I made my animation too big. You would think I'd learn after 400 or 500 times. All right, so let's, as I say, we want to learn a few of these basic signaling pathways, at least know something about the details. And students often groan as we go over this, but you need to keep in mind that over half of the metabolic pathways in all the cells of your body utilize this type of signaling. And many of the diseases, many of the pathologies that you will be working with at some level involve dysregulation of signaling pathways such as we're looking at here. Oftentimes it may be a genetic deficiency or an impaired expression, genetically or environmentally, of some component, either that we see here or downstream of this. All right, so I think we've seen this before. I think you know the general rules. I'm going to make sure because I can't tell you how many times on a test I've seen folks confuse the terms that are used to describe the components of this simple PKA pathway. What we're looking at here is the protein kinase A, the adenylate cyclase sequence. The red thing up here is the hormone. It's then going to bind to a G protein receptor. The R there, emphasis on receptor. That's not the G protein. That's the receptor that activates the G protein. The green, whatever color that is there. That's the G protein. Remember, the G protein is trimeric. We only see 2 subunits here, but there are 3 subunits in the primary G protein, alpha, beta, and gamma. And upon receptor binding, there's going to be a conformation in the G protein receptor, exciting dissociation of one component of the G protein. It's not always the alpha. In fact, even in the G protein cascades, that's not always the case, for reasons we won't get into. But when that happens, when there's dissociation, in, let's say in this case, the alpha subunit dissociates, and then it travels in the membrane, or in the interior of the membrane, to this large membrane protein called adenylate cyclase, or adenylyl cyclase, I don't care what you call it. It too changes conformation, and it's an enzyme that generally acts as a hydrolase, it acts as an ATPase, and it's going to release a product of ATP cleavage. But these are we'll look at that in more detail in just a moment. But these are the basic components of the G protein PKA pathway. I'm going to have to stop this one. I think this one includes the PLC pathway as well.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell. The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP within the cell.
So, to this point, has anything left the membrane in this animation? No. What about those purple things? What are they? Did he identify them? Yeah, those are the cyclic AMP. I can't get this thing. Here we go. There's a label there. All right. So in this illustration, we're going to call this our second messenger. Why? Because in this illustration, this is the molecule that actually leaves the membrane to bring information from the hormone binding to the interior of the cell. Usually that's true, not always.
The cell. The cyclic AMP then binds to and activates a target protein such as alpha kinase, which adds phosphates to specific proteins in the cell.
So a couple of things we want to make sure that we don't gloss over here. Where did the cyclic AMP come from? ATP, so adenosine triphosphate. All right, when it's cleaved, will yield adenosine monophosphate, cyclic adenosine monophosphate. So that's the origin of that. The adenylate cyclase up there in the membrane has an ATPase activity. It can hydrolyze the ATP. All right, and remember the other name for this pathway. It goes by a variety of names. In fact, sometimes it's called the cyclic AMP pathway. We called it the adenylate cyclase pathway. but it's also called protein kinase A pathway. So add all that together. And so what's this protein kinase A doing? It's just kicking off that intracellular activation sequence, phosphorylation sequence that we talked about before. Did you have a question?
Will the cleavage of ATP to cyclic AMP give off energy?
Yeah, and that's, the energy is sort of, got to be careful here as well. The energy remains latent within the transfer of that phosphate. So that energy is now being communicated to each of these monophosphates. Remember what we're doing in these kinase reactions, we're transferring phosphate from one molecule to another. The silly analogy I'm using is I'm setting something on fire or charging a battery, but we're spreading energy throughout the cell, enabling a lot of other reactions to occur. Yes?
You said that the cyclic AMP is the second messenger.
Yep.
How would you characterize the G protein knowledge? Is it?
No, the G protein is, I guess you could say it call it a membrane. It's a membrane that resides and never leaves the underside of that phospholipid bilayer. And while it's part of the second messenger signal transduction cascade, it would be erroneous to view it as the second messenger because it never leaves the membrane. It never conveys any information to the interior. Now, I will agree, I will concede as we move on to this next pathway that it is not always the case, that my little rule is not always true, but I think for the purposes of our discussion, it's a good first approximation. Whatever molecule leaves the membrane and carries the information to the interior of the cell will always be a second messenger, it just may not be the only second messenger.
The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated.
All right, so he's going to go on to the next pathway, and if I remember, I'll come back to that. But are there any questions then about that particular pathway? Darn it. So you can see the elements here. We can see our hormone, we can see our G protein receptor here, we can see our G protein. Again, don't confuse the two. You can see it's trimeric there. Here's our adenylate cyclase. Now, by the way, that adenylate cyclase in most textbook representations is called the effector molecule. I guess because it's really initiating the signaling process. It's releasing the cyclic AMP. And then here's our protein kinase A, which is going to utilize that phosphate to then phosphorylate. Again, remember, there are ATPs involved in all these steps. So this is an energetically very expensive signaling sequence. But we're transferring phosphates along this entire signal amplification sequence. Now, at the bottom here, I've sort of slipped in something very important, I guess because I was in a hurry, but I want to make sure you see what we're talking about. And remember, we said that it is generally the case that hydrophobic hormones act as prescription factors. That would apply to steroids and thyroid hormone. We've indicated that amines and peptides must bind at the membrane and therefore must utilize second messenger signaling sequences. However, it is very critical that you understand, very important that you understand that hydrophilic hormones can act as transcription factors. And one of the common pathways by which this is done is through second messenger signaling that culminates in the nucleus of the cell. The most common pathway for this to occur is called the Krebs pathway. It has nothing to do with Hans Krebs and the Krebs cycle, the tricarboxylic acid cycle. This is simply an abbreviation for cyclic AMP response element binding protein. And this is, the details of this pathway are far more complicated than we can get into here, but make sure you understand that through this Krebs pathway, and there are many other pathways, this is simply one of many pathways, a water-soluble hormone can modify or can result in or can cause the modification of intranuclear transcription. So again, just to be sure you understand all these components, here's another illustration for the quiz. Make sure you can identify the G protein, the components of the G protein. Again, the adenylyl cyclase is the effector. Here we can see the hydrolysis of the ATP, formation of cyclic AMP, sometimes called inorganic phosphate or organic phosphate over here. And then the protein kinase A, which is only activated by the cyclic AMP. All right, let's look at the next pathway. Remember that table that we looked at before sort of indicates why these are important, because a number of hormones, including a number of the hypothalamic hormones we're learning shortly, utilize this pathway. So very similar, we have a hormone binding to a G protein receptor. The G protein receptor changes conformation upon hormonal binding. The G protein then dissociates, causing a modification, in this case, not of an adenylyl cyclase protein. Remember, the adenylate cyclase is a transmembrane protein in the pKa pathway. Rather, this is a phospholipase. If you had to guess, what do you think a phospholipase does for a living? Right, it's an enzyme that catalyzes cleavage of phospholipids. And where do you find phospholipids? In the cell membrane. All right. And when certain, it's not all, but when this enzyme catalyzes the cleavage of certain membrane phospholipids, there are two primary products that we need to be familiar with. One, inositol triphosphate, we'll just call it IP3. The other, they don't show here. The other is something called, that you may remember from your basic biology, DAG, diacylglycerol. So these are the two cleavage products of the phospholipid, catalyzed by phospholipase C, or PLC. Either or both can act as second messengers. You can sort of see that in the little arrow here, because the information is leaving the membrane and passing to the interior of the cell. And then I think you see what's the consequence of the PLC pathway. What is the biological consequence of PLC activation? Dramatic. We're turning on that intracellular calcium switch. We're opening the ER. We're opening whatever the organelle is. It's not always the ER, but in this case, most commonly it is the ER. Liberating intra-organelle stores of calcium. And boy, we've already seen what can happen when calcium levels go up. Everything from contraction, smooth muscle, changes in cell adhesion molecules on the cell membrane, blood coagulation, you name it.
So you said IP3 and
DAG.
but then this one is calcium.
Yeah, that's what I was quibbling about before. Yes, remember my qualifying, the way I was going to back out of it, is I said that IP3 will still be a second messenger. It's just that in some cases, most of your textbooks will call this a second messenger too. I would call it more of a switch, but fine. I mean, if you want to tell me that calcium is a second messenger in the PLC pathway, that's fine. You should be aware that. But I think our rule, just to for me, it's a more formulaic and comprehensible recipe for understanding this process. In second messenger signaling, it's that second messenger which really takes the information from the membrane that's critical. Calcium's not doing that technically, but it does act as an intracellular switch. So I suppose on that level it's appropriate. And then I have, I think, another list here of all the things somewhere that calcium does. It'll come up shortly, but we've already talked at length about the importance of intracytosolic calcium concentrations. Why is it appropriate for me to say intracytosolic rather than intracellular? Yeah, because this is part of the intracellular environment, but it's this concentration that is exquisitely regulated by the cell through a variety of calcium binding proteins, pumps, and so on. Again, like the bilge pump we talked about earlier. And nature has all kinds of ways of fouling this up. Cholera toxin fouls up. You don't need to know the details, but cholera toxin fouls up the G protein pathway. Pertussis coughing. The pertussis toxin does a similar thing. So it's a very common site of nature's toxins. All right, the third of these signaling pathways, and perhaps a little bit simpler, is the tyrosine kinase pathway. I gave you a table earlier showing you some of the hormones that utilize the PKA pathway and the PLC pathway, and as I say, I'm more interested that you remember those as they come up in our discussion to follow. But this one's important, and I do want you to pay attention to the hormones that utilize this pathway now, because they're so critical. Most growth factors, virtually all growth factors, use some variant of tyrosine kinase signaling. Everything from the insulin-like growth factors, epidermal growth factors, platelet-derived growth factors, neurotrophic factors, they all utilize some variant of the tyrosine kinase signaling. Growth hormone itself uses a cousin to this pathway. It's a growth hormone. And finally, insulin also, at least at the outset, utilizes tyrosine kinase signaling. So it's important to know for that reason. You need to be aware of it, if only for those reasons. Now, we have several illustrations here. Here we have a little larger illustration. So unlike the very complex sequence that we've talked about in the previous two processes, this tyrosine kinase, which is it's a monomeric protein, a transmembrane protein. There's an extracellular component and an intracellular component that goes all the way through the membrane there. The extracellular component binds the hormone. In this case, it could be an epidermal growth factor, for example. And then as a consequence of that, remember we talked about these tyrosine residues being sites of phosphorylation. What tends to happen is that upon hormone receptor binding in the tyrosine kinase pathway, the hormone receptor complexes dimerize. Remember we talked about dimerization in the steroid pathway. They come together at the membrane. And then they engage in rather unspeakable acts. Excuse me, you're mature adults. Yeah. First of all, and it's not shown here, and I can understand why, just for reasons of decency. They autophosphorylate. Yes. They autophosphorylate. And then as they're autophosphorylating, as you can see here, they cross-phosphorylate. And this, in turn, again, that's why it's called, this is called intrinsic, it's not important, intrinsic tyrosine kinase, because this receptor has the ability to, again, to initiate this process on its own once dimerization has occurred. But again, and what's not shown here is all the ATP that's being sucked up in this process. And then furthermore, this phosphorylation cascade, or this phosphorylation event, is then transmitted to additional intracellular molecules, those molecules that are now going to be activated, and they in turn have the ability to amplify this signal intracellularly. But that's the general sequence of events, and I used to have a little video here. Let's see if that's still there.
Receptor tyrosine kinases exist as monomers free-floating in the cell membrane. Each receptor has an extracellular domain and an intracellular domain. When growth factor is present, it binds and induces dimerization of receptor monomers, resulting in receptor activation. The activated receptor then autophosphorylates tyrosines in its intracellular domain. The phosphotyrosines are binding sites for intracellular signaling proteins, which in turn can activate other downstream proteins. An exponential signal amplification occurs for each growth factor molecule binding the receptor tyrosine kinase.
So that's what is intended by, again, these terms that are used to characterize this pathway: dimerization, auto and cross phosphorylation. But the key thing is this excites intracellular phosphorylation, resulting in all those possible consequences we talked about before.
In the video, there was only one signal molecule. Is that possible with
So you're
just
asking,
one, or
no,
because
you
all
need,
of our images are showing two
yeah,
signal
you would,
molecules?
they would both have to be found. The dimerization can only occur when the conformational change is induced by the hormone binding. All right, so those are the three general pathways. Any questions then about the sequencing pathways, signaling pathways? All right, I also promised you a healthy dose of memorization, so let's begin that process. You need to memorize some hormones. To that end, let's talk about the hypothalamic pituitary axis. The hypothalamic pituitary axis. Do you have this slide? So I was looking at it this morning. I was impressed by its complexity because, but it's actually wrong. I was looking at it this morning. It's actually incorrect. You might want to go through it, if not now, as we continue our discussion of the inference system, so you can find out the errors. It's just something to do. I know you have a lot of leisure time.
Yes, that's right. Too much.
All right. So this is one of my introductory explicatory slides. So let me just talk through the main points here. We've indicated that the endocrine system primarily begins in the hypothalamus. All right, so the hypothalamus, unlike what the major magazines and tabloids would have you believe, is the central regulatory center of the endocrine system, not the pituitary gland, because the pituitary gland responds to whatever directions it receives from the hypothalamus. We will find that the hypothalamus releases specific hormones. These are called the hypothalamic releasing, or yeah, we'll just call them hypothalamic releasing hormones for now, that travel down to the pituitary gland, causing the anterior pituitary to release its own set of hormones. But you can see it all begins with the hypothalamus. So we call that the hypothalamic pituitary axis. And there are two general pathways by which the hypothalamus and the pituitary gland communicate. Number one is by a portal system. The portal system is just a system of adjacent little capillaries. The portal system is just a very small, tiny network of capillaries. And number two, by a tract, T-R-A-C-T, which you may have read back in the Reformation. Martin Luther. What is a tract? What is a tract? We talked about this. What's a tract? Like the corticospinal tract? Yeah, it's just a white matter connection. Yeah.
Dr. King, what was the first one? I'm sorry.
A portal system, which is just a capillary, tiny capillary system. The reason I emphasize that is folks so often confuse those two. I want to make darn sure at the beginning there's no possibility of confusion. All right. So those are the two general ways they communicate. Now, anatomically, you should be able to tell me, since we spent a lot of time talking about this in anatomy, anatomically, what is the avenue of communication between the hypothalamus and the pituitary?
Individual?
Yeah, the infundibulum, or the pituitary stalk. It's just a tiny little thread of tissue that connects the pituitary. Remember, it's seated down there in the Turkish saddle, the cella turcica. And the hypothalamus resides above that, down there close to those mammillary bodies and anterior to the brainstem. All right, so make sure you understand that basic organization, and we'll build upon that as we go. Now, I said a moment ago that the hypothalamus, when it releases its hormones, those hormones travel down to at least for the portal system, travel down to the anterior pituitary and cause the anterior pituitary to release another set of hormones. All right. Anytime a hormone causes release or excites release of another hormone, we say that hormone is trophic. So trophic hormones cause release of other hormones. And we won't get into the etymology. I think in one of the slides I talked a little bit about that, but that's the general definition. All right, some more terms. In your medical reading here after this course, you're often going to see reference to primary or secondary hormones Or the alternate terminology is central or peripheral hormones. So I want you to be aware of those terms. When we talk about and let's begin with secondary. A secondary hormone is a hormone that originates in the hypothalamus or the pituitary. A secondary hormone originates in the brain, with some exceptions, but that's the general rule. A primary hormone originates peripherally in the, I just gave you the answer to the other one. It originates, for example, in the thyroid gland, or the gonads, or the adrenal gland, pancreas. But you would agree, most of those tissues are not in the brain. Now, in the European vernacular, they prefer, of course, a lot of your reading will be with European findings. They prefer the term central and peripheral. So by that analogy, what would the central hormones be?
Secondary.
That's the secondary hormones. And the peripheral hormones are the primary hormones. So just be aware of that distinction. I'll use those terms quite a bit. See down here that most endocrine disorders, this is not a Panther Phys course, but most endocrine disorders originate or involve hypersecretion, hyposecretion, or impaired receptor sensitivity. So most endocrine problems mean there's too much hormone, and that could be everywhere from hypothalamic all the way down to the primary systems. Too little hormone. This happens, for example, in type 1 diabetes. What's the hormone that's missing there?
Insulin.
Yeah, insulin. Or it could be, there could be adequate amounts of primary and secondary hormone. There just may be an inability of the cells to respond to the hormone. And this is quite common too. This would be type 2 diabetes to some extent. There are a number of diseases you're going to talk about. Many other types of renal diabetes involve this type of problem. So here we're looking at that hypothalamic pituitary axis, and they stand anatomically. Here's our hypothalamus up here. Here's our infundibulum. Here's our pituitary gland. Now, note that we're sort of dividing the pituitary gland into two parts. There's an anterior part and a posterior part. You don't remember left or right because I can switch it on you. But we need to make sure we pay attention to the two, because the posterior pituitary is brain. It's actually neural tissue. The anterior pituitary is not. It's endocrine. And embryologically, they have very different origins. Again, while we're learning terms, in the medical literature, you will often see adjectives such as hypothesial. We're going to be talking about it here. Hypothesis, not hypothesis, but hypothesis. You should know that hypothesis means pituitary. The neurohypothesis is the posterior division. The adenohypothesis is the anterior portion of the pituitary gland. And you're going to see that quite frequently in the literature, because a lot of the drugs, in fact, begin with those terms.
What was the anterior one?
Adeno.
Oh, it's written here.
For example, in your reading later on, you're going to read about adenohypothecial tumors, which are, as I think I said before, present in about 1 in 20 of us, according to the research. And the other thing, we won't get into this in great detail. I have this more evident in the other slide. The hypothalamus is further anatomically divided into a variety of nuclei. Remember, the thalamus is also broken down into a variety of nuclei. You don't need to worry too much about the individual names, but you'll notice over here on the left, these nuclei are sometimes classified as mammillary, tuberal, supraoptic, and preoptic, and so on. And just remember all the things that the hypothalamus is doing, right? We talked about this before. Either through circuits, its connections with the autonomic nervous system or the endocrine system, and you can sort of see the putative division of labor up there at the top. The hypothalamus plays a role in regulating heart rate, vasomotor tone, digestion. By the way, we said there are critical centers in the brainstem that do these things. So how do you reconcile those two facts? Where do the brainstem centers in a lot of their information to make the decisions they make from the hypothalamus, right? Because the hypothalamus is monitoring a lot of information from the body, blood pressure, blood gas levels, hormone levels. But while I'm not going to ask you to remember these, to memorize these regions for the nuclei, why this is important is because a number of the cells we're going to be talking about, a number of the hypothalamic cells that are responsible either for releasing hormone or actually extending down into the neurohypophysis originate up here. So I just want you to be familiar with the general anatomy of this system. All of these neurons that we're going to be talking about have cell bodies up here, these hypothalamic neurons. It's just they go to different parts of the pituitary to different degrees. So the way that we're going to break this down in very simple terms, we're going to differentiate big and small cells in the hypothalamus. We're going to call them magnocellular and parvocellular. Magno's big, parvo is small. But these are our two fundamental classes of hypothalamic neurons. Now the big and small primarily refers to the length of the axon of those neurons. And I'm not sure it's entirely evident from this illustration, but you can see that some of these neurons, for example, extend all the way down through the pituitary stalk into the posterior pituitary gland. So they have cell bodies in the hypothalamus. There are the cell bodies up here. The axons extend all the way through the pituitary stalk, and then the axon terminals are located in the brain matter, the neural material of the posterior pituitary, down here. These neurons, these magnocellular neurons, release their hormones. These are neurons that release hormones. Again, we call that neurocrine. They release their hormones directly into the general circulation. And there are only two of those hormones, oxytocin and ADH. That's the easy part. The parvocellular neurons, and you can see these are small, these neurons are smaller and shorter. And where they terminate anatomically is not that critical, but what I want you to notice is they terminate at this proximal capillary system. If you can see in this illustration those purple blood vessels there, those are part of that portal system that we talked about earlier. Now you can see that portal system extends, we're going to say, from the pituitary stalk into the anterior pituitary gland. So the parvocellular neurons release their hypothalamic hormones into this micro capillary system, where they travel in that capillary system down to the anterior pituitary. So they travel from point 1 to point 2.
So in the middle is what they call the portal system?
Yeah, this whole thing right here, all those purple blood vessels you see, not these, but the ones that extend from the pituitary stalk into the anterior pituitary, that is called the hypothalamic hypophyseal portal system. The hypothalamic hypophyseal portal system. Remember earlier we differentiated the hypothalamic hypophyseal portal system from the hypothalamic hypophyseal tract? Remember that? I said there were two ways they communicate? All right, so don't confuse the tract. The yellow is the tract. That's just but the portal system is the system, this very, very tiny little microsystem of capillaries. You have several portal systems in your body, but this is the first one we'll talk about. So is that clear? And so the parvocellular neurons, and this is where it gets confusing for folks, and you may want to cogitate on this one. The parvocellular neurons are hypothalamic. They originate in hypothalamus. They release hypothalamic releasing hormones. That's what we're going to call them, with a few exceptions. That travel then. That's them traveling. They travel down to the anterior pituitary gland by that portal system, where they bind to receptors in the membranes, in the cell membranes of these sort of brownish cells here, which are not neurons. These are endocrine cells. And when the parvocellular hormones bind to receptors in the cell membranes of the endocrine cells, that causes the anterior pituitary cells to begin releasing their hormones. So do we all have a handle on the distinction between the hypothalamic hypophyseal portal tract or portal system and the hypothalamic hypophyseal tract? Make sure you understand the distinction. The tract is utilized by the magnocellular neurons. The portal system is utilized by the parvocellular neurons. What's a trophic hormone? A hormone that excites release of another hormone. So these hormones are going to be trophic. And so you have several illustrations. What are we looking at here? These cells up here that I've circled in red, are those magno or parvo? Parvo, everybody see that? So they're going to be releasing hypothalamic releasing hormones into the portal system. The portal system here is oversimplified, but I just want to make sure you see that that's what they're conveying here. They travel down to the anterior pituitary, exciting release of anterior pituitary hormones, and there will be several of those you need to remember as well.
So is only the parvocellular trophic?
Well, so excellent question. We're just going to define a trophic hormone as any hormone that excites release of another hormone. As we're going to see when we start labeling them beyond that, it gets a little more complicated, but certainly it is true that these hypothalamic, these parvocellular products, the hormones from the parvocellular neurons are categorically, incontrovertibly trophic. But there are some other hormones that are also going to be trophic. So that's pretty much it. We're going to have release of these. We're going to, because we already know ADH and oxytocin are pretty simple. That's a boring story, right? So we're primarily going to be focusing on these anterior pituitary. So here are our hypothalamic parvocellular neurons. They're releasing their hormones to the anterior pituitary, and then, as we've indicated, the anterior pituitary is going to release its own hormones. You're going to have to memorize these, including, for example, prolactin, growth hormone, thyroid stimulating hormone, adrenocorticotropin hormone, and the gonadotropins, FSH and LH. We say there are usually 6 of these. So these you got to memorize, and they all have different names, and that can be a little bit confounding. By the way, when they bind, let me go back up. Let me go to this first, and then we'll stop with this. But I want to make sure we get through this today. So in these tables, we have listed the parvocellular hypothalamic neuron hormones, and then here are the anterior pituitary hormones we just talked about, the 6 of them. All right. Now, look at those hypothalamic hormones. What do most of them have in their name? Yeah. So there's your tip-off. If the hormone has in its abbreviation an R, it almost certainly is a hypothalamic hormone, releasing hormone. Yeah.
Is it another name for somatostatin, GH?
Yeah, we'll talk about that. Okay. Yeah. There's a bunch of, we got to sort through that terminology later. Well, that's very confusing, so we'll come back to that. This is the preferred term now for a variety of reasons we'll talk about later. So I guess my directive, my imperative for you is make sure if you don't already have these hormones memorized, that's priority number 1. You just need to know those for the remainder of your careers. And we'll stop there.